So we're talking then about our new paper that we're putting out with a number of collaborators. We can talk about that important collaborative aspect perhaps a bit later on. So the paper is called the Genome-Wide Association Meta-Analysis of Spontaneous Coronary Artery Dissection Reveals Common Variants and Genes Related to Artery Integrity and Tissue-Mediated Coagulation. So what it means in reality is that what we've been doing is we've been working collaboratively internationally with a number of our friends and partners in order to try to understand better the common genetic variation which contributes to the risk of SCAD. So to explain this a little bit more, there are rare genes, and we've looked at some of these genes before in the work that we've done. These are the sort of genes that would run through a family. So you might have a single gene that was altered that led to disease, and then you would see the disease run through the family. Single gene disorders in SCAD do seem to be uncommon. They maybe occur in as few as 3 or 4% of all of the patients who present with SCAD. And those genes occur primarily in association with other established or known syndromes, other things that we know about, things like Marfan syndrome, Louis-Dietz syndrome, vascular Ehlers-Danlos, and adult polycystic kidney disease. So for most patients, the 96% of patients who have SCAD, it doesn't seem to be caused by a single gene mutation, a single gene that's gone bad, if you like. In most patients, it seems to be more to do with the common genes. So these are genes that we all have, but in SCAD patients, the hand that you're dealt of all of these different genes when you're born has a combination, which means that you have slightly more risk of having SCAD. And that's what this study was looking at. It was looking at those common genes, those risk genes, each of which maybe contributes a little bit of risk to having SCAD, and trying to understand what those were.
Can you tell us what genetic variants have already been identified as risk factors for SCAD?
There are a few genes that have been identified already. So we were involved with identifying the first common gene described in SCAD, which is called the FACT-R1 locus. These things aren't terribly memorable. But this is an area where there's variation in the genetics in the population. And this is a variant that's associated with the risk of SCAD, but also with other related things like carotid dissection, cervical dissection, fibromuscular dysplasia, and migraine. And this is a pattern that we're going to see a little bit more in the other genes that we're looking at. These genes also seem to relate to vascular diseases in other places. So the findings are very interesting and I think exciting from the perspective of our understanding of SCAD, what causes SCAD, and also opening up some routes for us to investigate and understand again a bit better why SCAD occurs in patients. What these genes show is, first of all, that quite a lot of the, if you like, the risk of SCAD is heritable. It's due to genes. For this reason, we were able to identify quite a lot of genes that conferred risk in SCAD. So there are 16 genes in total, some of which have been described before, as we said a moment or two ago. So 11 of those genes are being described in this paper for the first time as risk variants. So these are these little changes in these genes that are associated with SCAD.
So what are the new risk loci and what do they tell us in relation to SCAD?
So these genes there are a number of sort of common themes, if you like. First of all, a lot of the genes seem to be associated with the glue, if you like, that holds cells and tissues together, which we call the connective tissue. And the elements of the glue, so the proteins which stick things together, so the things that cause those cells to hold each other together or to bind to the bits of the connective tissue. This, of course, makes sense because we know that SCAD is a condition which is caused by a bruise which separates out the vessel wall to create this false lumen. If you remember, the tube carrying the blood and this bruise is making a false lumen by tracking along inside the wall of the artery. And so it makes a lot of sense that something that just makes those tissues stick together a little less firmly might make you more vulnerable to that. One of the other genes which is interesting and is novel to SCAD is a gene called tissue factor, which is involved in clotting, particularly within tissues, within the vessel wall itself, potentially here. And again, that makes a lot of sense. We know this is a bruise, so there's a clotting process involved, and therefore changes in how blood clots within tissues might make people more vulnerable. So I think the exciting and interesting elements are, first of all, that quite a bit of the risk of SCAD seems to be to do with genes. It has higher heritability. And actually, that's probably perhaps the reason why we've managed to identify so many genes. So in other conditions that are due to common variants, sometimes you need tens of thousands of patients to be able to identify one that causes a risk. In this study, we are able to show all of these genes that are relevant to SCAD from a smaller population of patients. So that's the first thing. The second thing is that the genes tell us something about how the disease works in terms of this, these aspects of how the connective tissue works and how it sticks to the cells and binds the vessel wall together, and the aspect of the clotting element. I think the other really interesting aspect is that how these genes that we've identified in SCAD interrelate to other diseases. So when we look at other vascular conditions, some of which have some association with SCAD, like intracerebral aneurysm, fibromuscular dysplasia, carotid dissections, migraine, we can see that the genes which have already been identified in association with these disorders, that the genes that we're identifying in SCAD overlap with these GWAS, the Genome-Wide Association study looking at all these genes. They overlap with the genes for SCAD. And interestingly, the conventional cause of heart attacks, that's ischemic heart disease, as we call it, or atherosclerotic coronary artery disease, also these genes are involved in that disease, but they are directionally opposite. So the genes which confer this risk of SCAD actually are slightly protective against the conventional form of coronary artery disease. So there are lots of interesting things that these genes are telling us about, if you like, the underlying vulnerability in SCAD patients. And of course, what that does is it opens up a whole avenue of research questions and routes to enable us to kind of probe this further and try to understand how we might be able to modify that risk to the benefit of patients.
Do you think these findings will help to move SCAD research forward?
Yes. So I think that these genes tell us a lot about the next direction of travel. They're not the whole story. There will be, they if you like, they're the start. As I mentioned to you before, you learn more in genetics with these kind of conditions, the more patients you can get. So there's opportunity to build the size of the analytical pool of patient population. And again, I should say to all of the patients who have participated in the research, anybody who gave a blood sample will have supported this research. And also many volunteers who participated in other research studies. For example, a lot of the controls in the UK come from the UK Biobank. And all of those people that gave a little bit of their time, a little bit of their blood, have enabled this to happen. So yes, clearly going from really no knowledge of the genes which confer vulnerability to SCAD a couple of 3 years ago to now having 16, and not just sort of 16 random and separate genes, but 16 genes that are telling us a theme about what's going on, gives us a lot of insight. It allows us to start to understand more clearly what the vulnerability is that underlies SCAD. I think we've got a lot of further questions to go to work out exactly, from what the gene is and what the gene is doing to what's actually happening in the coronary artery. There are still big questions to answer about the other sources of vulnerability, so particularly the role of female sex hormones in SCAD. Still, we don't quite understand how that interacts with the genes. The genes are not, clearly telling us how that works. So plenty still to do, but what this does really for the first time is give us some clear avenues to go down, and some, actually at a molecular genetic level, some very specific targets that we can start to think about in terms of, understanding risk, and potentially starting to think about what might be treatment strategies that could be effective. And also, as I've said to you, these genes are kind of conferring risk to a bunch of vascular diseases, but also protecting against ischemic heart disease. So what we've got to be thinking about with our SCAD patients is we don't want to be kind of making one thing better and the other thing worse, or whatever it is, if you see what I mean. So it shows you the complexity of how these diseases work, but it gives us, it gives us for the first time avenues to follow to really start to understand the underlying causes of SCAD, which is one of the critical, obviously, probably the most critical question for the research.
Is there anything in these findings that support medication decisions, for example, in controlling blood pressure?
That is, again, one of the key findings that emerges from this study, which is that when we look at conventional cardiovascular risk factors in SCAD patients, and this is something that we've noticed clinically, we don't tend to see an increase in cardiovascular risk factors compared to the general population. Whereas obviously in patients with conventional coronary artery disease, we'll see more smokers, more people with diabetes, high blood pressure, high cholesterol, and so on. But the exception for that seems to be blood pressure. And when we look at the genetics, this is looking at the genetic predisposition to these cardiovascular risk factors. High blood pressure does come out as being the genes which confer a risk of blood pressure seem to be associated with the risk of SCAD overall. And what that's telling us is that blood pressure is probably an important factor that we should be considering in our management. We have to be careful that we don't go too far from genes to treatments, but I think it is also been suggested in some previous observational data produced by the Vancouver group that control of blood pressure may be important. Now, this isn't going to be in everybody, right? So there are quite a lot of our SCAD patients who have quite low blood pressures to start with, and so we have to be pragmatic. We don't want to be taking people's blood pressures down so they feel faint, dizzy, and can't get out of bed and all of those kind of things. But I think in patients who have high blood pressure or higher blood pressure, then this is a sort of an additional piece of evidence in that jigsaw that suggests that making sure your blood pressure is well controlled if you're a patient who has high blood pressure and SCAD is probably a sensible thing to do.
Many SCAD patients say they were emotionally stressed before their SCAD. Do you see any links between stress and a genetic predisposition to high blood pressure?
I think we probably have to be cautious about inferring too much in that particular direction. I think we do know that emotional stress is important in SCAD patients, particularly in female SCAD patients. I also think clinically we have to be careful about sort of overstating that in a sense. I'm always conscious of the sort of stress spiral. If you get stressed, worrying that the stress is going to somehow cause something bad to happen can be quite stressful, and then you can get even more stress and you just end up in a sort of an escalating spiral of stress. So yes, I think stress has a role. Exactly how that interacts with these genes, I think, we're a way off being able to make all of those connections. But what we are starting to be able to do, which I think is the point that you're making, is to build a clearer patchwork of what confers risk in SCAD patients. So we've got a bunch of genes that confer risk. Not everyone's going to have these genes. As I've said, they're common genes, the genes in the population. So it's not going to be a question of identifying, your genes and then, that's an easy solution to you. But they tell us something about what confers risk. Emotional stress tells us a little bit about what confers risk. High blood pressure, that does feed a little bit into the genetic findings as well, but blood pressure is obviously relevant to some patients, again, not all patients. But probably getting this cumulative risk, and in some patients it may lean more in one direction as being important, in others in another direction, but we're building a clearer picture of what makes up the vulnerability that leads to SCAD occurring in some patients.
The paper talks about there being a high polygenic heritability for SCAD. Does that support the current thinking that family members of SCAD patients don't need to have genetic testing?
Yes. So the polygenetic bit of that is what I was talking about in terms of the common genetic variation. So the genes that we're talking about here are not, all genes run in families, right? So all of your genes, if you have children, your genes will pass, half of your genes will pass to your children. But these are not single gene disorders in terms of the fact that you have an abnormal gene which then runs through the family. These are common genes. We all have them. I have these genes, and I may well have some of these variants. But it's the combination of these variants. It's, as I said, it's the hand that you're dealt when you're born that gives you that vulnerability. Now, what this high heritability is telling us is that in terms of the risk of SCAD, the proportion of the risk which seems to be due to these common genes is an important contribution. It's not the whole story, but it's an important one, and it may be that it's more important than in some other diseases where the role of genes is smaller, if that makes sense. So these genes are important. So again, what that's telling us is that this is a good thread to follow in terms of understanding the underlying causes of SCAD and getting a better grip on things which might have an impact in terms of patient care.
Where is the paper being published, and how important is it to the SCAD world?
The paper is going to be published in Nature Genetics, which is a very, very high profile journal, and I think reflects both the quality of the work that's been done by our international partners, and particularly our collaborators in France, who have been doing a lot of the analytical work behind the work in this paper, led by Nabila Bouatier-Nadji. So, again, I think what we should say is a thank you to Beat SCAD for all of the fundraising that has been done by many, many people doing different things, from baking a cake to running a marathon to whatever it is. I know all sorts of wacky things, but all of those wacky things add up to us being able to extract DNA from blood samples, do genetic sequencing or genetic analyses to allow for this work. And, I think one of the exciting things about the paper, not just where it's going in this very prestigious journal, but also that I think it has something to say, not just about SCAD, but about vascular diseases and arterial diseases and how they all interrelate to each other as a whole. And, you know, I think it will be of interest to many scientists, not just those of us who are working and keen to try to improve outcomes in SCAD patients.
What questions does the study raise, and what are the next steps for research?
So I think that these things are interesting questions. And, you know, what these things are telling us is that these genes and changes in how these genes work and affect how the vessel wall works can manifest in a bunch of different ways. In some patients, it might be SCAD, in other patients, FMD or migraine and so on. And obviously what we need to understand now is kind of almost like the next step. So we can see that there's these genes that confer risk to a bunch of vascular conditions. So why is it one patient gets SCAD and one patient gets migraine, or one patient gets SCAD and migraine? But maybe, do you see what I mean? We have to try and understand that. And, you know, I think one of the other questions is that, you know, genes gives us an understanding of risk and vulnerability, if that makes sense. But it doesn't tell us why SCAD happens at a particular point, because that vulnerability has been there since you were born. And so, you know, we still need to understand what it is about that day that makes the SCAD happen at that time. And there are obviously all sorts of elements to that which may be interesting. We talked about emotional stress, we talked about female sex hormones relevant to women, and maybe more exercise related things, or, you know, who knows, maybe there are things that we don't yet know about. But I guess if we understand more about the vulnerability, it also helps us a little bit, because we can start to potentially build models in cells and so on that allow us to try to understand how, if you are vulnerable, what are the triggers, and how do the triggers work that cause the SCAD to happen at that time. So there's obviously more research to do. But I think it is reasonable to say that thanks to SCAD patients, Beat SCAD, and obviously international fundraisers, our international partners, this is a big step forward. And I think I should also say one very important thing, which is that doing a genome-wide association study, which is what GWAS stands for, is something that no individual research group in the world can do in something like SCAD, because it's relatively uncommon. Certainly not at the moment. So the only reason that we've been able to do this work is because of the amazing collaboration of our partners internationally, France, in Australia, in New York, Michigan, the Mayo Clinic in Vancouver. And, you know, we should just say very publicly our thanks to all of those groups for agreeing to participate in this study, which, as I say, we think is a real stepwise change in our understanding of the underlying vulnerabilities in SCAD patients.